everyone, I welcome back you guys to my channel. I hope you all are doing fine, must be studying and enjoying your life. So as you have seen the thumbnail, today I will cover the topic of diatom examination, forensic examination of diatom. Why are they of so much importance when it comes to death by drowning? In order to know ante-mortem or post-mortem drowning, how is it possible that this minute structure could be helpful in order to recognize things and to identify the culprit? So if you want to get answers of all those questions, you should be there with me throughout the lecture. And do not forget to subscribe our channel because I will be posting lectures like this. So let us get started. First of all, you must know what are diatoms, because the whole story, the whole scenario, we are trying to focus on them. So it is very important for us to know some basic details about this species. Diatoms are nothing but microscopic and photosynthetic and unicellular algae. They belong to the class of Bacillariophyceae. So in forensic science, they serve as the biological marker for drowning in most of the cases, or I should say in almost of the cases, because their cell wall are composed of silica. Silica is hydrated silicon dioxide. Then they are nearly indestructible. They don't decay easily. They don't get destroyed by strong acids easily. Now moving on to the anatomy and classification. I hope you understood what are diatoms. So let us have a look at how do they actually look like. Their anatomy is basically divided into two major portions. First is look the anatomy or the frustule portion. The diatom cell is encased in a very unique glass shell. This glass shell is nothing but frustule. The valvar structure comprises of two overlapping halves, just like the soap box, similar to a petri dish. The larger or the outer portion is epitheca, whereas the smaller, which is the innermost portion, is hypotheca. Ornamentation is also there, like the surface of the frustule is covered in patterns of pores, that is known as pancha, ribs, that is known as costae, and lines that are known as striae. These patterns are very unique to each species. Then the classification of the diatoms. They are basically classified as centric diatom, that are known as centrales, and the pinnate diatom, that are known as pinnales. Now what are centrales? They have radial symmetry. They can be circular, they can be triangular. They are most common in marine environment and in stagnant water. Stagnant water in stagnant lakes means which is stable, which is not moving. Then pinnate diatoms are also known as pinnales. They have bilateral symmetry. They are elongated and at times they can be boat shaped as well. They are dominant type in freshwater sources, be it river, be it streams, be it pond. Now some protocols and tests that are involved with diatom as an evidence. First thing that a forensic expert is desired to be done is sample collection. The femur or the thigh bone is most preferred sample in case of death due to drowning, in order to identify whether it is post-mortem or ante-mortem. Fine. It is a closed system. Since the diatom cannot naturally exist inside the bone marrow, finding them there proves that they were carried by the bloodstream, and if blood circulation was there, then there are super high chances that person must be alive at the time of death. Now we have acid digestion method, the most important method when it comes to testing of the diatom. To see diatom, the organic human tissue must be destroyed. First of all, the marrow or tissue is placed in a flask. Now which reagent is used? HNO3, concentrated nitric acid. The mixture is boiled and the acid digests all the protein and lipid whatsoever present. Then the silica frustule remain intact, because as I have said before, it is quite hard to digest it. Because glass does not dissolve easily in nitric acid as well, and this could also be one of the reason. Then we carry out centrifugation along with the microscopy. The digested liquid is spun in centrifuge and the heavy silica settled down somewhere at the bottom. This sediment is then placed on the SEM, that is scanning electron microscopic slide, or any of the high powered light microscope. Now what is the purpose? In order to count and identify the species species of diatom that might be present there. Fine. So this was the test, this was the examination that is carried out in the lab. Now something most important, whether the drowning was ante-mortem or post-mortem. Ante-mortem drowning, what's that? Means person must be alive when pushed inside the water. Post-mortem drowning, person was already dead and the dead body was thrown inside the water. So uh, here's then a beautiful image which I hope is quite clear, but I will explain this to you. Wait. In case of ante-mortem drowning, active inhalation of water occur. Why? Because person was alive at the time of death. But in post-mortem in post-mortem drowning or post-mortem submersion, it is post-mortem submersion actually. Passive entry of water into airways occur, but no active inhalation. When it comes to the circulatory system portion, heart must be still beating in ante-mortem drowning. Diatom enter the blood via ruptured alveoli, but in post-mortem drowning or post-mortem submersion, there is no circulation, no movement beyond the lung. Now, when we talk about the diatom location, it is found in closed bones or closed organs, especially in bone marrow, liver, brain, in case of ante-mortem drowning. But in post-mortem submersion case, it is found only in the lung but absent in rest of the part of the body. Why? Because no circulation was there. Fine. So here's the difference. Circulation was there in ante-mortem and diatom are there in the bone marrow, but in post-mortem submersion, no circulation. Okay. So no diatom present in the bone marrow. Now let us have a look at the real life case scenario, which is also known as the mismatch mystery. In this case, a body was discovered in the stagnant city pond. Some local police assumed that it might be a accidental fall, but the findings that were observed in this case were absolute game changer. Water sample was collected. The pond water, it contained only centric diatom, but what pathology report confirmed doesn't match this. The victim's bone marrow was filled with pinnate diatom. Imagine that. The area, the water body where he was found contained only centric diatom, but the pathology report confirmed that it had pinnate diatom. So the analysis proved that the pinnate diatoms are only found in fast moving freshwater stream, but the body was found in stagnant pond. What was the verdict? It proved that the diatom mismatch. It proved that the victim did not drown in the pond. Investigation further revealed that the victim had been drowned in a mountain stream which was 20 miles away, and the body has later transported to the city pond to mislead the investigator. So here the diatom acted as a biological GPS in order to solve the murder mystery. So I hope you got a clear-cut view that how diatom can be useful when it comes to tackling the drowning cases, when it comes to the mystery of ante-mortem and post-mortem drowning. Now let us understand the pathophysiology, means how the diatom enter the body. There are basically three phases via which the diatom's entry into the body can be easily described. First is the gasp reflex. When the person drowns in the water, they experience the terminal gasping, followed by alveolar or capillary membrane reflex. It occurs when the membrane is somewhat 0.2 to 0.6 millimeter thick. Then under the mechanical stress of drowning, it tears it, allowing the diatom to pass from the lung to the pulmonary vein. Just imagine how painful the process of drowning must be for the person. Then finally the journey begins. From pulmonary vein, the diatom go into the left ventricle of the heart, then the aorta, and finally into the systemic circulation. From here, it will transport everywhere in the body, like said before, in the closed organ, in the bone marrow, in the liver, in the brain, all the closed organ, because circulation was still there. It is the case of ante-mortem drowning. So this is how things occur. Here are some of the important note points for you in order to summarize it. The frustule is silica shell of diatom. The silicosis. What is silicosis? The inhalation of silica. This process is silicosis. Then forensic limnology. The study of freshwater biology, or the study of diatom is forensic limnology. Then alveolar capillary membrane, I have used this word. The barrier diatom must cross in order to enter the bloodstream. These all keywords are of core important. And you can also watch the lecture that I have previously made on the diatom. It is an excellent lecture, and I have compiled some handwritten notes in them as well. So do not forget to subscribe the channel, and thank you so much for being with us and watching the lecture.